So a very warm welcome to all of you guys. In this video tutorial we will going to talk about EIEC. We have talked about EPEC, ETEC, EAEC and EHEC in earlier video. This video will going to be the last video where we have talked about a strain of E. coli. From the next video we will going to talk about treatments, and we have also talked about diagnosis, right? So we have only one video left where we will going to talk about treatment. So let's start today's video where we will going to talk about EIEC. So Enteroinvasive E. coli is the full form of EIEC. Okay, so as its name suggests, invasive, that means they have the ability to invade their target cell and causing infection. Okay, so these enteroinvasive E. coli are biochemically, genetically and pathogenically closely related to Shigella. Okay, that is why they are also known as Shigella-like E. coli strain. Okay, but that does not mean that they will produce Shiga toxin. Okay, so they does not produce Shiga toxin. So whenever in MCQ part they will told you that which of the E. coli strain have the ability to produce Shiga-like toxin, the answer should be one and only EHEC. Okay, and if they are talked about name a E. coli strain which is Shigella-like, then you can talk about EIEC. Okay, so I have told you that they are invasive, that means they penetrate and multiply within epithelial cell. So epithelial cell, which is mainly the intestinal epithelial cell, is the target cell for EIEC. Okay, so they have the capacity to invade interstitial epithelial cell in vivo, and they also have the ability to penetrate HeLa cells in tissue culture. Okay, so that is the important point for MCQ. Okay, they can ask you that in vivo condition which type of cell is penetrated by EIEC. Your answer should be intestinal or interstitial epithelial cell, and in case of tissue culture they can also penetrate HeLa cell. Okay, so now upon invasion of the intestinal epithelial cell, they causes lysis of the phagosomal vacuole. Okay, so phagosome, where phagosome is present inside the macrophage, right? So phagosomal vacuole lysis allow them to spread through the cytoplasm and they infect adjacent cell. Okay, and Shigella also does the same thing. Clinically, EIEC infection, so as they are following Shigella in their mode of action, that means it is obvious the symptom will be similar like Shigella. So as here we are observing mild diarrhea, frank dysentery to severe inflammation and fever. Okay, so they are having non-fimbrial adhesins, intimin types adhesin. Okay, and they are in mainly possibly outer membrane. Okay, so we all know that E. coli is a gram-negative bacteria, so there are outer membrane present, and in the outer membrane there should be the non-fimbrial adhesin, and it is quite common, right? And in their plasmid there is a gene for K surface antigen. Okay, that means in their serotype K antigen serotype should be present. Okay, and they are usually non-motile and they do not ferment lactose or ferment it late with only acid production and do not form lysine decarboxylase. Okay, and that is why diagnosis of EIEC becomes quite difficult, because if they will ferment lactose readily, then we can easily use McConkey's agar to diagnose them, but here it is a quite difficult because they usually do not ferment lactose or ferment it late. Okay, with only acid production, and also they do not form any kind of lysine decarboxylase. So many of these show O antigen cross reactivity with Shigella. I hope you know about what is cross reactivity. Cross reactivity is a immunological term where suppose we have prepared a antibody for O antigen of Shigella. So instead of binding with O antigen of Shigella, they can also bind with the EIEC. Okay, so that is the phenomenon called cross reactivity. Okay, where antibody can also bind with the similar epitopes instead of binding with their identical epitopes. Okay, so that is the concept of enteroinvasive E. coli. What things you should remember, that enteroinvasive E. coli are similar to Shigella species, but that does not mean that they will going to produce Shiga toxin, but rest of the mode of action are exactly same. As their name suggests, they are invasive, that means they have the ability to invade their target cell. So their target cell here is intestinal epithelial cell. Upon invasion of the intestinal epithelial cell, they will lyse the phagosomal vacuole and spread through the cytoplasm and infect adjacent cell. That is their mode of action. Okay, and this mode of action ultimately causes mild diarrhea to frank dysentery, severe fever and other inflammations. Okay, severe inflammation, fever. Okay, so they are having non-fibrillar adhesin and they have a possibly outer membrane protein, and they have K surface antigen on their plasmid that they have acquired. So it is obvious that this plasmid is the pathogenicity island, right? Because they have acquired this, and that's it. And they are non-motile and they do not ferment lactose or ferment it late. Okay, and they do not form any kind of lysine decarboxylase that create a problem in their diagnosis. Many of these show O antigen cross reactivity with Shigella. So this is the pathogenicity of Shigella, EIEC. So what is going on? They will go via transcytosis, then they will lyse the phagosome. As you can see, they have escaped from phagosome, and from after the escape they will spread through the cytoplasm and infect adjacent cell. That is the idea. Although this detailed diagram or illustration is not necessary for us, and I have taken this image from the Nature magazine. So that is all about EIEC. Okay, so if we try to recapitulate it, so first of all we should tell that they are invasive bacteria, that they will invade the target cell and causes multiplication, and that is why we are getting symptoms, right? So their target cell is here intestinal epithelial cell. Okay, upon invasion they will lyse the phagosomal vacuole and spread through cytoplasm to adjacent cells. That is the idea. Okay, and they follow the same mechanism just like our Shigella, but they do not produce any kind of Shiga toxin. Remember, symptoms are mild diarrhea, frank dysentery, severe inflammation and fever. Okay, and another important thing, they have K antigen encoding gene in their plasmid. Okay, and they may show cross reactivity. Many of these show O antigen cross reactivity with Shigella, and I have explained what is the thing. Many of them are non-motile and they do not ferment lactose or ferment it late with only acid production, and they do not form any kind of lysine decarboxylase. So that is the total idea of enteroinvasive E. coli. I hope this video will be helpful to you. So thank you for listening to this class. From the next video we will going to talk about the treatment. Okay, so bye bye.